Hi, so my name is Justin, and today I'm going to be telling you about a project that I did with Rebecca, David, and Scott at Auburn University, looking at larval development and ecology in the shrimp Halocaridina. So in lots of organisms, larvae and the young undergo development in a different habitat than they will come to live in as adults. In aquatic and marine organisms, this is often called larval transport or larval export. One good example of this is in coral reef fishes. The larvae often undergo development in nursery habitats like mangrove swamps, and then as adults they'll return to live out the rest of their lives back on the reef. Another really good example of this is in the shrimp family Anchialine, which is what we're going to be talking about today. In these shrimp, the adults are strictly limited to fresh water. They're intolerant of seawater. But in many species, the larvae actually require seawater to develop into adults. So in these species, the larvae are exported into the seawater, and the adults then migrate back upstream, sometimes hundreds of miles, to live out the rest of their lives. Now there's a big difference in crustaceans between what happens physiologically when they're in seawater versus in freshwater. I won't go into the details of this, but essentially in freshwater they use energy to maintain an internal salinity that's higher than the surrounding freshwater. In seawater they don't use energy. They're called osmoconformers, and their internal salinity is usually the same as the external seawater. But there's one group of Anchiads called the Anchialine clade, which is shown at the bottom of this phylogeny, where the adults can actually tolerate seawater. And today we're going to be talking about one member in that clade, Halocaridina. And the reason that these shrimp are called, they're in this Anchialine clade, is because they're found in anchialine habitats. And anchialine habitats are basically these landlocked but coastal ponds, pools, caves that have no surface connection to the open ocean, but are connected to the oceanic freshwater as well as the oceanic seawater as well as the underground freshwater via all these little subterranean cracks and fissures and things like that. So because of these connections, the salinity in these habitats can vary quite a bit due to tides, and also depending on where the shrimp is at in the environment. And because of that, it's thought that these anchialine shrimp in the family Anchialine are able to tolerate different salinities than the strictly stream Anchiads. So in Halocaridina, a previous study that we did showed that not only are these guys osmoregulators, but they're among the strongest described osmoregulators in all of crustaceans. So they're able to tolerate a really wide range of salinities while keeping their internal salinity at a very narrow range. But there's this mystery surrounding Halocaridina that has to do with its larvae. So the adults are found pretty abundantly in anchialine ponds in Hawaii, but the larvae have never been found in the surface water of these ponds. And it's thought that the larvae are confined to the underground portion, sort of the cracks and crevices of these habitats. And so we thought maybe one reason that they might be confined to this underground or hypogeal habitat is because the underground waters in these habitats are much saltier. So they're usually approaching seawater, and they also have a much more constant salinity. So maybe the adults are able to osmoregulate fairly well, but the larvae are not able to osmoregulate, which is why they're confined to this underground salty and constant salinity environment. So to test this hypothesis, we took adults that we had in the lab. We waited for them to get pregnant. We then took larvae, early stage larvae from these adults, and split them into 3 different treatments: a seawater, brackish water, and a freshwater group. And we then monitored their development. And in the seawater and the brackish water group, we got high settlement rates, so 70 to 90% of these treatments, or these individuals in these treatments developed into adults, whereas in the freshwater group we got very low settlement rates, much lower than in the other groups. So I think this is good evidence for, again, showing the lack of larvae to tolerate a wide range of salinities. And to get at why that might be, we looked at the gills of the larva. So the gills are the primary site of ion transport and osmoregulation in crustaceans. These are some pictures of gills from the adults. You can see they're stained heavily here with this stain that stains mitochondria. And basically, I won't go into why we did that, but these green cells are the ones that are undergoing ion transport and are responsible for osmoregulation. So we wanted to look at the development of gills and of these cells during larval development. And you can see here that in the early stage larva, D here is the earliest and G here is the latest. We don't see any gills, and certainly not any ion transporting gills. In fact, we didn't begin to see gills until after settlement, so when they were young adults, and we started to see these little proto-gills forming. You can see that they're quite a bit different than the adult gills. They don't have those long, high surface area lamellae, but again, we don't see gills developing until the adult stages, providing an explanation for why the larva might not be able to osmoregulate, might not be able to tolerate a wide range of salinities. We also used the data set that we had been accumulating for about 5 years in the lab, that we were basically monitoring reproductive output in this species on a weekly basis over this 5-year period. And we did this to address two questions. Number one, is there any difference between genetic lineages in this species? So in Halocaridina, there are at least 8 different, very distinct genetic lineages, as you can tell from this map. And we have representatives of all these in the lab, so we wanted to see if there was any difference in reproductive output. And we also wanted to see if there was any difference due to seasonality. And you can tell that, sure enough, some of the lineages reproduced all the time, whereas others reproduced very rarely. And we also saw a peak in reproduction in the winter and the spring, and a drop-off in reproduction in the summer and the fall. So we think we've provided some interesting insights into Anchialine larval ecology, specifically why, or one reason why we might see lots of adults in the surface waters, whereas the larvae are confined to these underground waters, again, probably because they're not able to withstand the changing salinities in the fresher waters of the surface waters, because they can't osmoregulate because their gills don't develop until they become adults. And then also, again, we've just provided some sort of nice reproductive data on this species, which wasn't known before. So if you're interested, read the paper and let me know what you think. Thanks a lot.